Now this is part 2 of the introduction to genetics, focusing on Gregor Mendel, the father of genetics. So he was born in 1822 and was around from till 1884. He was an Austrian monk, born to a peasant family in Heisendorf in the Czech Republic on July 22nd, 1822. Gregor Mendel was considered an excellent student in high school. He studied philosophy for several years, and in 1843 he was admitted to the Augustinian monastery of St. Thomas in Brno. Here he took the name of Gregor and received important support for his studies and research. Here's an example of the Austrian monastery where Mendel was, an example of the Punnett Square that we will talk about. In 1849, Gregor Mendel was relieved of his pastoral duties and accepted a teaching appointment. From 1851 to 1853, he attended the University of Vienna to study physics and botany. He was sent to the University of Vienna for a teaching certificate. He did not pass the exams because he was too nervous and was not considered a clever student. In 1854, he returned to Brno, where he performed his first pea hybridization experiment in 1856. The garden pea is what Mendel is famous for, so take note of his experiments with the garden pea. He continued to research until 1864, when he became the abbot of the monastery. Some did not appreciate the work that Mendel was doing because he did not focus completely on the religious side, and as Mendel was interested in the work of Charles Darwin, so not the direct connection that the church perhaps wanted to Charles Darwin and the idea of natural selection. In 1884, Gregor Mendel died of kidney disease. The new abbot burned his papers, and I want you to think for a moment, why would he burn the papers? So think back to his interest in Charles Darwin. He was working with garden peas. He was looking at genetics. It perhaps was not the first priority of the church at that time period. Now, a local newspaper said this: "His death deprives the poor of a benefactor, and mankind at large of a man of the noblest character, and one who is a warm friend and a promoter of the natural sciences and an exemplary priest." Mendel's work was not understood nor appreciated while he was alive, and it wasn't until the 1900s and even into the 1920s and 30s when people truly understood what he had discovered. So there's a memorial monument to Gregor Mendel. Now, Mendel's experiments. He conducted his experiments with Pisum sativum, the garden pea. Now, he wasn't the first scientist to study the garden pea, but he was the one who distinguished himself from others through two main points. He had very careful planning, and he adhered to the scientific method. His work again was largely ignored by others until Correns de Vries, Steinmark, and Bateson all came up with the same ideas. So here's an example of the garden pea that Mendel was dealing with. Now, Mendel selected the garden pea for several key reasons. First of all, it was cheap and readily available as a seed in a variety of shapes and colors. Now, cheap is important because he works in a monastery, so there's not a lot of money to be spent on science experiments, and it was readily available. Peas normally self-pollinate. It's easy to cross-pollinate pea plants, so it allowed for his genetic experiments. It takes up very little space because, again, space in a monastery would be hard to come by. Garden peas have relatively short generation time. So within a single season, you can grow the plant, they will flower, they will produce peas that you can use, that we would eat, for example, he used for scientific experiments. Produces many offspring, so one pod would equal 6 to 10 peas. Now, considering the time, it's important to remember that Gregor Mendel was conducting these experiments when there was no knowledge of chromosomes or the role and mechanisms of meiosis. So they had no idea about cell division, how chromosomes moved within the nucleus of a cell before a cell was about to divide. So it's amazing that he came up with these results without having that information. Now, regardless, Mendel was still able to find distinct units of inheritance existed, and then he used this information to predict the behavior of gametes during their formation. So he kept accurate records and used that information to help him formulate his ideas. Now, he selected certain characteristics when he dealt with the garden pea, and first of all, he dealt with flower color. So, for example, in the picture here, you have white, but it also comes in purple, which may be referred to as violet. The flower position is either axial or terminal, so either at the end, which would be terminal, or axial, which would be further in on a branch of the pea plant. The stem length of the plant, they can either be a tall or dwarf referring to a short pea plant. The pod shape is either inflated or constricted. Now, a constricted pea pod would look something like when you're in the garden and you're trying to shell peas and you get one where it looks like the pea pod is vacuum packed onto the peas. That would be constricted, inflated are the pods that pop nicely. Now, seed shape, they're either round seeds or wrinkled seeds, and the seed color can be yellow or green. The pod color, on the other hand, can be yellow or green. Now, for each characteristic of the 7, flower color, flower position, stem length, pod shape, seed color, and pod color, each of them has a contrasting characteristics. So this will be the gene that Mendel references, and this will be the alleles or the options that we'll talk about shortly. Now, all of his plants were considered true breeding pea seeds. That means when it's true breeding, they will reflect exactly what the parent looks like. So white flowers, if crossed with white flowers, will give rise to white flowers. Each genetic trait would remain unchanged generation after generation. Now, you'll notice any of the keywords that we need should be in red here. So trait is basically another way to say characteristic. And a trait is a characteristic that is heritable from one generation to the next, so it's something that can be passed on. Now here's an example of the different types of parts that are characters that Mendel looked at. So he looked at flower color, so here's the example of the flower. Here's the flower position, terminal or axial, seed color, shape, inflated versus constricted, green versus yellow, tall versus short. Again, just another picture of some of the characteristics or traits that Mendel looked at and what would happen if we crossed a round with a wrinkled. Now, Mendel's experiments were successful because he chose garden peas, which was fortunate. He restricted his study to a few contrasting traits at a time, so he didn't try to study everything at once. He focused on just a few areas. He kept accurate quantitative records, so he kept track of his numbers. His analysis of the data allowed for certain postulates to be developed, which provided the underlying basis for transmission genetics. So because he kept good, detailed data, he was able to come up with some basic ideas that I'm still teaching you today.